Today, we're going to talk about cancer immunology and cancer immunotherapy. This is an area with very, very fast technology development. And as a result, a lot of new computational methods are developed around cancer immunology and immunotherapy. In the earlier lectures for this module, we discussed cancer mutation analysis. You can see a lot of the pediatric tumors have low mutation rate, but in solid tumors or tumors that occur facing the outside tissues or organs, or have onset later in life, usually they have much higher mutation rate. Right? So when you have very high mutation rate, the cancer genome is unstable. And therefore, even if there are mutations with a targeted therapy, very often these tumors can develop resistance very quickly. And so using a single agent to treat a cancer with many mutations is like just having one policeman to clear up this big traffic jam mess. The solution for this is really cancer immunotherapy. It was ranked by Science magazine in 2013 as the breakthrough of the year. So at the end of every year, Science would publish one issue to rank the top 10 breakthrough of the previous year. And in December, this is the breakthrough of the year in 2013 is cancer immunotherapy. And in 2018, James Allison and Tasuku Honjo won the Nobel Prize for their pioneering work in developing cancer immunotherapy. So in order to really understand cancer immunotherapy, we need to understand some basic cancer immunology. We know that in a lot of experiments, people grow human tumors in mice. And so a question for the audience is, would tumor grow in another individual? So can you take somebody's tumor to grow in another individual if you can grow tumor in mice? The answer is no, because most of the time when people are growing tumors in mice, these are immune compromised mice. They have very bad immune system. Normally, our immune system is able to recognize what is self and what is not self. And so if there are mutations, or even if no mutations, every individual, we have genetic differences. For example, SNPs that are different between individuals. And so if you have a particular tumor implanted in another individual or animal, the immune system of the host will reject the implant. And so the same situation when healthy cells create mutations, the immune system is able to recognize this as non-self and eliminate that. So this is called immunosurveillance. Our immune system keep on checking whether there are mutations in the genome. And if there are, it's very quickly recognized as non-self and eliminated. And so normal immune surveillance, you eliminate those foreign cells. Sometimes because immune system can be weaker over time. For example, the T cells in our immune system are the primary surveillance cell type. And it has receptors on the cell surface to recognize, to check, make sure everything is normal. But if there are mutations, the T cells can actually recognize this and eliminate the tumor. But then after puberty, our ability to generate different T cells or to recognize mutations are reducing gradually. And so sometimes there would be a mutation that happened, and the T cells that recognize this particular mutation might have died already in the body, and there's no new T cells replenishing it. Then a particular cancer cell could stay in the body in quiescence for a very long time. And this is called equilibrium. When people are looking at tumor mutation and evaluating the rate of mutation and the actual mutation they observe in the tumors, sometimes they observe early mutations that appear 10, 20, even 30 years before a particular cancer onset. And so some of these mutations happen early, and somehow immune system was not able to recognize it. And those cells are allowed to be there and just grow initially slowly. This is called equilibrium. And when the cancer cell really accumulated enough mutations and start to grow much, much faster, that's when you can detect the cancer. That's when the immune system has failed, and that's an escape. And so one early approved immunotherapy is called active immunotherapy. These are general reagents that just overall boost the patient immune system. And one important one is cytokines. So these are proteins that are secreted by our cells when, for example, immune cell encounter foreign particles. And so if you suspect somebody have a weaker immune system, you can just give this individual a cytokine. For example, IL-2 is one such immune stimulatory cytokine. And if you give the individual the cytokine, overall, they'll have all the immune cells will get activated. You can see here with the IL-2, it will stimulate the T cells, B cells, NK cells, and monocytes, just have an overall activation, which includes inflammation, differentiation, response, repair, just like overall boost. There are also immune suppressive cytokines and also immune active cytokines. So for immunotherapy, you usually want to use an immune active cytokine. It has been FDA approved, but only used in very small occasions. This is because this type of therapy has pretty bad side effect. If you think about COVID, it's because the patient immune system is too active, and sometimes the side effect could kill the patient. And so to activate the immune system without specifically targeting the cancer cells can be a dangerous thing. And so what really got immunotherapy to be widely used in cancer treatment is immune checkpoint blockade, which really only specifically target cancer cells. So we mentioned before that T cells in our body are the main immunotherapy kind of surveillance cell types. And on the T cells, there are receptors in here, which is able to distinguish self from non-self. And so you can imagine them as guards in our body. And all the normal cells in our body regularly present degraded proteins onto the cell surface through this MHC complex. And this is like all the normal individuals, they carry a passport and or they present their things on the cell surface, and the guards, the T cells, will just check to make sure, okay, this is self, this is normal, everything looks okay. But if the cancer cell has a mutation, there would be proteins that gets presented onto the cell surface, and the T cells can very quickly recognize, I have never seen this protein before, this seems to be a foreign thing. Initially, the immune system was evolved not really to recognize tumor mutations. This was mostly evolved to recognize different infections from pathogens, viral infection, bacterial infection. Those kind of will present foreign proteins onto the cell surface, and the T cell will say this cell got infected by some virus or bacteria. And so the reaction for the T cells when it sees a foreign antigen presented onto the cell surface is to actually kill the cancer cells or the infected cells. Unfortunately, with immune cell during the equilibrium or early evasion, what's going to happen is that the cancer cell will secrete these checkpoint genes onto the cell surface. These are cell surface proteins which can interact with the T cell, other receptors on T cells. You can imagine this is like passing either bribery money or kind of alcohol to the T cells to say, hey, buddy, we're friends, no reason to get alarmed. And so very often in the tumors, you will see that so with this interaction, the T cell function will be suppressed. And so in the tumors, you might see T cells right adjacent to the cancer cells, but they are not doing the killing job. And so immunotherapy, currently the really common one is anti-PD-1 or anti-PD-L1 antibody. They're also anti-CTLA4 antibody. Basically, these are antibodies which can block the PD-L1 and PD-1 interaction, which allow the T cells to be reactivated or rejuvenated. It's like you pour cold water onto the drunken soldier and they will just wake up and be back on the job again. And then very quickly they recognize, oh, there's just a cancer cell with a mutation, and it will kill the cancer cells. And that's kind of how current immunotherapy works. And this has really been used in many, many cancer indications in widely used cancer treatment.